Excellent. Great, thank you. Okay, yes, so like Jenny said, I'm going to talk about the ClinGen Allele Registry today, and hopefully provide some context for why that's useful and helpful for biocurators. And so yes, I'm the lead product owner for ClinGen from Baylor College of Medicine's perspective, and I'm the Bioinformatics Research Laboratory led by Alex. Okay, so I'd like to start with an overview of the topics that I'm going to cover today. So the first thing we're going to do is talk about the overview of the ClinGen Allele Registry, defining the problem and the attributes of the ClinGen Allele Registry that allow it to solve some of these problems, specifically for ClinGen, but also some outside groups as well. I would like to address the current status of the registry, along with the users and uses. Then I'd like to get into the existing functionality, mostly supporting the variant pathogenicity interpretation for small variants. Talk about the variant naming. I'd like to do a demo, talk about the API as well. I'd like to spend some time on the structural variant support and the plans, our existing support for CNVs, as well as what we have coming down the road with non-CNVs. And then next steps, and then summarize everything. Okay. So defining the problem, purview of genetic variation. So the goal of the whole genome shotgunning, I'm sure everyone's aware, the sequencing is to interrogate all forms of variation. And so we've essentially broken these down into these two main groups in how we're actually storing this data on our end and serving it up to the community in terms of single nucleotide variants or these small insertions, deletions, indels, and structural variants. So you see the separation between two of these in terms of how we structure them and how people can query them and register them in the Allele Registry. And so started a while ago with 1,000 genome projects, observing 4.1 to 5 million sites affecting roughly 20 million bases of sequences. They observed that most of these were SNVs and indels, 99%, but they also found around 2,500 structural variants per genome. Some of the more recent studies, and this is not an overview or a catalog of this, is a lot of things that happen in between this, but just in terms of the data scope and scale that we're looking at, we see roughly 800,000 short variants that are usable from the low millions of sites from these studies of the large scale, but also an increase of up to 27,000 structural variants. So you see that the scale of what we're able to get on the data end versus what we're actually able to store within the Allele Registry. So in terms of ClinGen, where this started with us, in terms of at least the registry efforts, so NIH developed the ClinGen, the Clinical Genome Research Program, of course, to develop these growing needs of enabling testing, clinical testing labs, researchers, patients, and providers to share data associated with systematic implementation of standards for critically relevant genome variation, as it relates to genetic testing, variant classification, and data exchange. The items that we're mostly going to be talking about today is some of this curated knowledge and how we're able to use variant naming to power some of that, and variant pathogenicity, how our efforts are to support this. So the solution for the ClinGen Allele Registry, we aim to support the stable and consistent variant identification and naming, which is curation for pathogenicity efforts. So this is one of the FDA requirements for classification process. And the end result of at least the short variants is producing these canonical allele identifiers, these CAIDs. So you'll be seeing this a lot. And this is our naming convention for these short variants. We also wanted to support open sharing of genetic variant information, aggregation of information from different sources, and all within these FAIR principles. So definable, accessible, interoperable, and reusable. Also wanted to be able to produce identifiers on demand, so high bandwidth and low latency, so not have a bottleneck of a long process. So people getting identifiers from ClinVar and dbSNP, they issue variant identifiers, of course, but it's a completion of a more robust process. So R, on the other hand, is accepting any valid identifier, any valid expression to create the canonical allele identifier. So we also provide API support for programmatic registration and access. In fact, everything that's possible to do on the UI with the Allele Registry is accomplished via the APIs. And so everything you can also do on the programmatic end, and I'm going to show an example of that. So it also supports large batch queries and registrations. And also we provide this JSON output for programmatic integration with other tool sets. And the last bit was free and open user registration. We will make it very easy for people to get registered, create these logins that allow them to register canonical alleles. and no login is required just for viewing and surfing. Okay, so here's the manuscript that I believe that was sent out in the email. So this is first authored by Piotr here. And so this basically, it gives a breakdown of the structure of the Allele Registry and the initial population of this data. The main points I wanted to discuss here is that there are these different concepts of which you'll see on the canonical allele identifier page, which I'll be showing an example of shortly, that's related to the genomic contextual allele as well as the transcript contextual allele. And that allows us to get this nucleotide reference from the nucleotide reference sequence, but we also have the effect that actually is the protein contextual allele, the change on the protein end. And this example below here, you can see the genomic allele specifically for GRCH37, 38, and NCBI 36, and the HGS expressions associated with those. So talk a little about the use, where we are right now at the higher level before we get into some of these details. So over the course, the last 4 or 5 years, we've seen some different spikes in registered variants, and this is the canonical allele. So these are CAIDs that we have available that have been registered. And a lot of these spikes are associated with the release of external data sources, which I'll show example of a couple of those in a little bit. But the main points here is that we're over 1.1 billion canonical alleles right now in the Allele Registry. And the average API registration rate or querying rate is right around 50,000 a second. So, for example, we were able to get, I was able to get Nomad 2.0 in roughly about 7 hours. And so it's pretty fast. It's written in C++, so it's pretty optimized on the back end to allow us to do these high bandwidth registrations. And some of these parts of this graph look flat, but I did want to have an asterisk here letting you know that this is roughly about 122,000 new variants over the course of this time. It just looks flat compared to the global scale of everything. So here are some of the registry usage with the users and frequency. So we see right around about 20 unique users or institutes register canonical alleles a month. And we've seen that number grow over the course of the last couple of years. We see roughly over 250,000 visits to landing page. We see about 14,000 UI uses a month from about 100 countries and about 1 million API uses a month from a little over 25 countries. And you see our trends. We have some spikes periodically with some groups doing some bulk querying and some bulk registration. So we see some spikes periodically, but our growth has continued to grow in the last couple of years. So I'm going to talk about the usage, some of the tools that are using this before we get into some hands-on examples and do some demos and some interaction. So we've seen canonical allele identifier adoption from various databases, including ClinVar, Civic, myvariant.info, Ensemble, database of pathogenic variants, and others. The tools that use this currently are Link Data Hub, Variation Curation Interface, Evidence Repository, Functional Data Repository, and some other tools as well. And some data aggregation that we want to mention. So we're getting some more use from a couple of these answers here. All of us clinical laboratories are identified variants classified by more than 2 laboratories. So it's allowed them to get basically doing a naming convention for their variants. The VIC was able to integrate across 6 prominent somatic cancer variant knowledge bases to register variants and aggregate variant interpretations. We also saw something very similar from the Australian genomics platform, Shariant, did the same thing across everything. So we've did a lot of different groups within and outside of ClinGen use this with some success. So one more point before I get into some of the more illustrative items of this talk. So this is built on these reference databases, so from NCBI 36, GRCH 37, GRCH 38. Roughly over 700,000 transcripts currently exist within the Allele Registry that are mapped between these different reference builds. And we do a monthly update on that. And we also annotate with some additional items. And one thing I want to mention was using these matched annotations from NCBI and Ensemble using this main select identifiers. Okay, and so phrasing the problem of what we're trying to solve and how we're going to solve that is looking at these different variant curation challenges. And the main point here we want to bring up is that variant identity. So seeing the same variant being defined differently in different sources. And I'm sure everyone here has seen across different manuscripts, finding different representations of variants, different identifiers, and trying to figure out, are they talking about the same thing or not? Also, diversity of evidence sources. trying to pull in as much data as possible for curation purposes. There's also, a lack of standard ways to accumulate this evidence. So getting everything into one centralized place for dissemination has proved useful. It's also important for tracking the provenance of evidence using interpretation. So we do this with the Link Data Hub, which I'll talk about towards the end of the talk. Also, when they have transparency and auditability, which is key FDA requirements, and alert users and curators to knowledge and content changes, very important. So these are some of the concerns in Allele registries that will address some of these specifically. Okay. So this evidence aggregation, we see this as a three-step process, and we're mostly going to focus on this first step, is actually getting these entities registered. And then from once you get an identifier, you can then link to external searches using that same identifier, and then excerpting additional data elements using that same identifier. So it allows us to do this linking process to empower downstream tools and users. So the Allele registry is mostly populated with these large external databases that we've collaborated with, including myvariant.info, Cosmic, ClinVar, Nomad, Exec, DBVAR, and a few others. And this allows us to populate a database and allow people to describe what they witnessed other things and get this canonical allele identifier that allows them to use additional tools and link data across different platforms. Okay. So here's an example of a canonical identifier. For those familiar or not familiar with this, you can see we have in orange here a genomic HTVS for the 3 builds for GRC 38, 37, and NCBI 36. We also have the ClinVar variation ID. We have the DB SNP ID. We have my variant identifiers for HT19 and 38. We also have these ClinVar records. We also have the Nomad identifiers, and you'll see a lot of these transcript HTVSs for this particular canonical allele. So all of these changes happen to be, some of them in the same location, some of them are a different location, depending on the transcript, whether it be NCBI and ensemble. And you see that they all have the same effect of the C to T. So this is what it looks like within the Allele registry, and we're going to do a live demo a little bit to kind of show you what these various bits look like live. But you can see from this small example here, this network shrunk down, you can see where this different data resides. So at the very top, we have the identifier and link outs to different resources, and these are different links. So if someone gets to a canonical allele from any of these various HTVS expression, VCF, type of inputs, they can easily get to these external data sources, and I'll show an example of that in a second. We also have the genomic alleles in this middle section, and below that we have the transcript alleles. And you'll see this orange label here for main preferred, which main select, that lets you know which with the matching annotations between NCBI and ensemble. And one note here is that this happens to be, this UI is, started up in HTML format, but everything is also available in JSON. And I'll show it, and you could look at this JSON from this link here, but also show an example of that. Okay. Before I get into a use case, Jenny, did you have any questions or anything before I get actually to a live part of the talk?
I don't see any questions in the chat, Kevin. Does anybody have any questions? Feel free to put them in the chat or unmute yourself if you have any questions.
Excellent. Thank you.
Right, thanks.
Okay. So continuing, I'd like to use a use case to kind of maybe more of as a jumping off point to, I think, the most pertinent bits of why biocurators should be interested in the Allele registry. So I found a paper kind of doing a reverse search. I started off with an expression and then found a paper that had that. So this paper just chosen by happenstance, but it has listed here some, it says these following common cancer somatic mutations. So what I wanted to do is show how someone could start with a manuscript and actually work their way backwards using the Allele registry to actually determine if something is already registered in the Allele registry, and if it's not, how would one go about registering that so they can actually, do some other downstream work in the face of curations. So the HGS expressions were not published. So let's say whether they were or not in this paper, let's say they were unavailable. We only have a G name, a location, and effect. Okay. And so the question is, do we have existing C canonical allele identifiers for these variants? So we can start off going to the Allele registry, which it's reg.clinicalgenome.org. And so we can start by figuring out if these are already in here. So I'll make this a little bit larger. Here we go. Okay. So we can start off, and I'm going to show you some of these search options after the fact, but I wanted to start off doing a more interesting part in terms of how it relates to the Biocurator aspect. So the search here, it says, do not have a transcript or HGVS expression. It allows you to start with a gene and the variation-based query. So I'm going to click on this. And so we're going to use this example here. So I'm going to start with TP53 and 743. Okay. And before I click on this, so you'll have two options here. One is actually for the transcript, which is the C notation, which is from the HGVS nomenclature, or you have this P nomenclature, which is the protein. And I want to show examples of both of these. So if we start off with the transcript option, I see that the reference is G. Let's put that in here. And the alternate is A. So this is a way of looking for this query if you don't have the HGVS expression. And so we hit query here, and it'll take a couple seconds. It really depends on how large the gene is. Okay, and we get our answer. And our answer is that, yes, this has been identified. And this is our canonical allele identifier, CA000387. It's on ClinGen 17, of course. And you also see all these helpful link outs in this search page result. Let's click on this and actually see what is included on each one of these pages before we do a couple more searches. So you see that as we talked about earlier, we have a canonical identifier at the top. We also have the gene name, and we have link outs to HENC and then CBI. Within here, we have link outs to the ClinVar RCVs, as well as dbSNP, exact, nomad, cosmic, myvariant.info. And we also have some additional links from Civic, eRepo, and PubMed that are related to this specific identifier. And so these are all link outs if anyone's interested in finding out more information about each one of these. You can click on these and it'll take you to the more in-depth information that's outside of the Allele Registry preview. Additionally, we have this JSON LD. And this is more for the programmatic individuals that actually want to interoperate with the Allele Registry. And you can see here, we basically have everything in this JSON linked data format where we have all these external records and these link outs within here, as long as they're identifiers, followed by the genomic alleles. The HGVS is for each one of these. So you can see each one of these are here is GRH 38, 37. And then at the very bottom is you have all your transcript information. And all that information, most of it is also on this UI page. But for people who are interested in doing things at a more automated fashion, you can use this JSON. And so let's say that someone wanted to do some curations based on this, and they were collecting different papers. They could actually start off, using, going to the VCI or other tool sets. You can start off with this identifier that may have had all these different types of representations. This could have been listed in a paper. This could have been listed in a paper. And these different representations, especially with things with different coordinates that are a little more ambiguous, in Ensemble and CBI, you now have an identifier in which to work with. Let's look at another example. Let's say you had a protein effect in this paper, and I'm going to start off with this graph gene as well. 1799. In this case, we're starting off with the protein. Let me see the analytical. Okay, so we'll see if this is registered. Oop. Putting the wrong symbol there. One second. There we go. Sorry, the wrong window here. Here we go. Is it clear? And so we see that this is also already registered as well on the chromosome 7. And so you can see an example of that here. In this case, it has very similar information in these same data sets as Cosmic does. Sorry, Cosmic is one of them, but as the TP53 variant had. I wanted to show one example of one that does not exist, and to show people how they can actually go about registering one of these variants. So same thing, we start off with generating this. So I'm going to start off with this crash gene. we'll say C. Actually, sorry, one different effect here. I'm going to show actually an indel. Shown. If you click this button here, it's going to switch over to indels instead of an actual SNV. If you click on this, it'll allow you to do the same type of thing. So I'm going to click on crash here. I'm going to say it's encoding, and I'm going to say 35 to 37. They're going to be a deletion. And so you can query this to see if it exists. It shouldn't. Okay. So it says it's a valid allele, but it's not present in the registry. So what you have the option to do here is to register this allele. And if you click on this, it should then register this allele. So what I'm going to do is I'm going to save this HGVS expression, just so that it's registered after the fact. So now we see that.
Sorry, can I ask, how did you choose? So you chose just like the top one, but there were a bunch of different options. So that's where I get confused a lot of times.
That's a very good question. So it's best if the manuscript has that information. If the manuscript at least tells you what the base name is for the transcript, and from there you generally have the option to say that is it likely the same change in the different transcripts. If they don't have it, people in the past have contacted authors to say, can you provide us with the transcript or what the identifier that you used at the time? And chances are of the 700,000, the Allele Registry may have that. The worst case scenario is having to guess, to say that, oh, I have the option of 3 transcripts and they disagree with what is at that location. Sometimes you have to make an educated guess, but that's a very good question.
Yeah, and this is Jenny. So I see for this one though, the HG38 is the same. Excuse me, it's the same for all of them. So that would suggest it's the same genomic change, but just having, a different effect on different transcripts, I guess. So it should be okay. But it does bring up a really great question with the fuzzy query. I mean, sometimes you will see more than one CAID that comes up, and then which do you choose? And I don't know what to do with that. That's a tricky one. But just to make sure people are aware of that and look out for it. Are you really only seeing one variant or more than one?
Yeah, so I think that I think that it's something we can talk about with the VCI and having some way to indicate that, we've registered it, but it's actually just our best estimate of what it would be after we've, tried to contact, et cetera. So that'd be good.
No, very, very good points. And yeah, and I think that's the best way to proceed forward. Sometimes you can do enough kind of internet sleuthing to figure out what it likely is, but sometimes, you just have to contact the original authors. And so that's essentially some of the tools that allow people to get from a lot of the manuscript type of expressions that they provide to getting canonical allele identifiers. However, the best case is actually having HGVS expressions, which I'm going to talk about in some more detail in the second part of this demo. So if we start at the beginning, I'm going to use that example that I copied from that class that I just registered, and expect to see this long number. And so you can see that these numbers represent pretty much close to the number of canonical alleles that we actually have. So this looks like 1.139 billion, which is pretty close to the real number, because everything essentially goes sequentially. And so for this drop-down query, we have different options, and I'm going to focus on the short nucleotide, the short variants, or the single nucleotide variants first, before we actually start talking about the copy number stuff in the second part of this talk. So in this case, we have an example of the search-based HGVS description, HGVS expression, or a descriptor. I'm going to do the HGVS first. We'll do the descriptor in the second part. So this is the HGVS that we had just registered by doing the fuzzy query and seeing that it was not registered. And so if I do a search based on this, I expect to get that same number. Yes, and it is. Let's also say, for this case, and while we're here, let's say that this manuscript, it didn't refer to this, but it's an example. Let's say they also said, it described this position 35 to 38 was also a deletion. And let's do a search based on that. It does not exist. So if you're logged in, you can click Get Identifier, and I expect it to be registered. So this should be probably close to that same number. It happens to be one more. It might not always be sequential, but you see that as one more now. So this is now registered. For people who do not have a registration already, it's simple. If you just go to the reg.genome.network or reg.clinicalgenome.network, it's towards the bottom. We have an option to log in. I'm already logged in, so you don't have that. Let me show you real fast. Log out, log in. So if you see this create login, or if you forgot your login. So some people might forget their username or their password. So as long as you remember what email you have registered, you can do that, or you can always create another account or contact us if you have any problems. Okay, so I'm going to go back to the main page. I'm going to show you some additional query options. So you can also query by the canonical allele identifier. It should take you to the page as you'd expect to see. You can also do based on some various things. I'm not going to go through each one of these. I'll show a couple. So the ClinVar variation ID should take you to the same thing. I'll do one more of these. Let's see, like Nomad, for example. And these are basically these different types of identifiers that should take you to their respective canonical allele page if it does exist. Yes. Okay. You also have the option of searching based on the gene symbol to get a full listing of everything that we have for that gene within the genomic coordinates. So again, depending on the size of the gene, some of these queries will take a couple seconds, but that was relatively fast. And you can also see in this payload up here that you could also tweak the URL yourself. You can bookmark these things. Everything we do is more or less exposed via these URLs. It's based on how we had set up our APIs. So you can see that we have TP53. It says skip 0. That means start at the very first one and limit it to 100. And so this is the result page for the first 100 variants for TP53. And you scroll down, you can see these asterisks here before the gene starts and before the transcription starts. And so you see the very bottom, you also have, you can go do this next functionality or previous if you're already on an additional page. So you can increase this limit to size that would eventually time out if you wanted 500 or something like that, or if you wanted the second 100, or if you wanted to have bookmark some of these things, you could say skip 500 and give me 10. You can do these different types of things. So it lets you know where you are globally as well at the bottom. We also allow people to search based on reference sequence and position. So this is looking at a particular reference. Instead of starting with the gene, you can talk about a particular reference and look at the position within this reference. So you can see here from these locations on this gene, you have the first, in this case, only 36 results. Okay, you can also search based on PMID, PubMed ID, and I'll talk about the copying stuff in a little bit. And we already talked about the genome variation-based query. There's one more I wanted to show, was interactively generating this variation. So let's start off with an example, just see if we can get the same thing back. So let's start with using this TP53 and see if we can get that same identifier that we got earlier. So if you slowly type, it'll give you the available gene dropdown. So if you only know part of it, or you want to see what else is available using similar strings, you can start with that. And then from here, it'll allow you to start off with the reference that you're interested with. A lot of people will use like the main select transcript. And then we have a link out to the actual sequence that was used as well. So this is at position 743. So we can find that. Let's see, it's a line number. Sometimes it's a little tricky, but you can kind of guess. Let's see, previous one, probably in the middle. Yeah, 733. I was off at one. So 743, it's a G, and now it's an A. So if we do a search for this, yeah, it pulls up that same number. I recognize that 387 from before. So there's different ways to get to see, if a canonical allele is available, and if it's not available, it'll give you the option to register it. That's the main functionality for these short nucleotide variants. As well, yeah, so you also can see this get identifier part. I'm not going to spend a lot of time talking about the APIs, but you can see from these different paths. So for example, we talked about where the JSON existed. So this is serving up that JSON directly, but we can also do these things from the command line, and people are interested, and just show as a real simple example of that. So if I just do a curl, which is basically like, you know, obtaining this URL, it'll give you that same JSON information. You could output that to you know, a different tool, a file. People could do bulk uploads, bulk downloads using these types of utilities. So I want to mention about the APIs. Okay, there's a couple recent APIs additions, and I would mainly want to bring these up as how they might affect users of this working group. So we're working on extending these different queries. And then we're eventually going to add these to the UI. One thing is querying the alleles, specifying the sequence name directly. And you can see this NM here versus having to do the genomic coordinates. And another one is actually just saying chromosome 17, for example. And, you know, if you use GRCH38 by default. We're trying to add these as people, you know, have different information from manuscripts, and they're collecting these interesting variants, and they're having trouble, you know, finding out if they're in Allele Registry or not, based on, you know, limited sets of search queries. So we're constantly trying to add additional functionality to help people, and so feedback is always welcome as if something's missing that might help people. Okay. Does anyone have any questions about short variants before I jump into structural variants? Or actually, I'll ask Jenny if there's anything in the chat.
Yeah, actually, one point that was brought up was just whether it might be helpful to add the single amino acid code to the 3-letter amino acid code. I guess, for example, when you're using the fuzzy query, the gene variant-based query. So that might be something that's helpful for folks. So that was just one comment. I do have a quick question. So when you have the list of, you know, all the different possible, you know, transcripts and where that variant falls, some of them have that VV in the little blue box is that verified variant? And can you maybe just give us a bit of detail on what that means? Yeah, that's it.
Yeah, so it's variant validator. I think they changed, I don't know how recently this was, but I think they changed their endpoint, because the last time I looked at this, it threw an exception. So it was essentially trying to do is actually making sure that, you know, these variants that are listed, you know, they're actually accepted by different protocols and different standards. But what we need to do is to figure out if it's just a small URL change, or if we need to serve up something different.
Okay, so
hopefully I'll have an update on that soon.
Okay, thanks. So if it doesn't have the VV next to it, should we be more cautious about that? Is that what that means? No,
I wouldn't. So I think that the rule is basically, I think that they initially accepted, and I'll have to double check on this, that they initially accepted just NM transcripts, you know, from RefSeq, and not, you know, the XM or anything from Ensemble. Okay. But I need to verify. Yeah.
Okay, great. Thank you. So any other questions at this point before Kevin moves on? Okay, great, thanks.
Okay. I'd like to talk about structural variants, and I want to make sure I save some time towards the end, so I don't want to get into too many of these things, so much just with the functionality. But the overview for structural variants and talk about some of the complexities that basically change between the way we're able to handle these short variants versus these more, you know, the structural variants and complex variants. So the tools and resources for structural variants lag behind the capabilities, you know, the sequencing technology, processing pipelines, models, naming conventions that exist for smaller variants. Probably because of a lot of times it was the data that was being generated. So on average, structural variants are still very, very important. You know, 2.9 rare occurrences that, you know, alter coding regions that they basically affect the dosage or structure. So we've received an increased interest in actually, you know, registering these and naming these and finding some utility to actually, you know, get an identifier that people can use in downstream curation efforts. So they account for about 17.2% of rare alleles genome-wide. So I think that's always seen, you know, recent increase from some of these manuscripts. 90% are non-coding deletions, three times more likely to associate with GWAS study than SMVs. And as these large SVs are allowed to, they're likely to affect the expression of a gene, like compared to an SNV. So yeah, again, these things, challenge the researchers and really looking for these disease-causing variants, in these unresolved Mendelian disorders. So detection has it definitely improved, and that's resulted in multiple useful databases. And ClinVar, DBVAR, Nomad, Exact, and others have actually started categorizing these, and releasing these catalogs as well as some rough structures that that naming conventions that they're using, whether it be extending VCF or other things like that. So for us, there exists a real good dereferenceable naming convention which identify these single SVs across genome build and transcript versions, which we'd like to do like we've done with the SNVs in the Allele Registry, as well as the small variants. So stage one is this, we worked on this copy number variant, structural variant support in the Allele Registry, which is started in 2019. And the stage two is this non-CNV structural variant support in the Allele Registry. And I'm going to show you some examples of those in a couple of slides. So our goal is to essentially extend the Allele Registry to support naming registration, querying of structural variants in addition to existing short variant support. So we want this to be a centralized resource for supporting clinical WGS interpretation, serve the needs of ClinGen tools and users first, and then go to the larger community after that. And then we would like to devise a generic model that allow for additional types down the road. So this is a basic overview of structural variants, and and this is driven by, I guess, the publication itself from listed here at the bottom of how we view these. And we don't have to get into all these details, but I wanted to kind of bring these up as a precursor to the what is driving our descriptor model, what we're actually creating on our end. So the way that we're looking at this right now is these structural variants. They're basically variants that are generally, 50 base pairs and upwards to millions of base pairs. This 50 is arbitrary. It's changed seemingly a bunch of times, but fine. We can kind of use that as a basic model. But what we're using to drive this, I say, more than the size is the source of the location. So if Nomad has a structural variant release, when we're treating them as structural variants. So for copy number variants and structural variants that are types of deletions, duplications of the genome, and then say, okay, well then, what about structural variants versus complementary variants? Well, CNVs are unbalanced structural variants with gain or loss of genomic material, often detected with read depth analysis. And so common formats we see in the VCF and BED. This is another way of looking at this. Looking at this canonical structural variants. So the CNVs appear at the top and the non-CNVs in the middle, and then complex at the bottom. So if we kind of fast forward a little bit, to say like what we're currently supporting is in this green part of this pie chart here. At least for the initial release of Nomad, we saw that CNVs are almost 66%. Non-CNVs are about 32, almost 33%, and the complex is small, but definitely present. So this is the CNV descriptor that was derived from ISCN, ClinVar preferred name, and some other sources that allow us to get the ball rolling. Because other groups that are actively working on this, GA4GH and others, are actively working on modeling what these structural variants look like at the very detailed level. And our thoughts is that we want to adopt these models as they become available. And initially getting off the ground, we started off with this descriptor that allows us to use this Rango database to actually list them and actually do some searches and queries and registration of these things. So you'll see here in this descriptor for CNVs, we have the assembly, a chromosome, an interval, which can be precise or a range at either end, and then its dosage. That allows us to specifically work with multiple copies. So this is a registration summary where we're currently at. The main point here is that we, compared to the 1.13 billion short variants that we have in the Allele Registry, the CAIDs, we have roughly 344,000 CACN IDs. And we have, we're continually expanding these endpoints that allow people to actually get some of these summary data. And so we're working on doing something similar for the Allele Registry at a smaller end. Copy this link. Let's see if you can sample what this looks like. So this is a JSON structured format. It shows you for the CNVs, not very big, there we go. What assemblies we support, the 3 different assemblies, and then the counts at the very end. So right now, yeah, we have this 344,688 entries. And then it also breaks down CMV counts per build. So you can see exactly where we're populated, and we want to make this a little more enriched. And so when people are curious of what we have and where we sourced it from, we're going to keep extending these types of functionalities via these API and REST endpoints. Okay, so I'd like to show you a couple examples of the existing queries that we have. So the first one is descriptor. So we already talked about the HTTPS descriptor, but this is the descriptor that I just referred to. So if you do a search on this, hopefully it'll bring up a CACN ID, which is similar to the CAID, but we use a different prefix to kind of separate things out and allow us to categorize different things. And you'll see that similar, we don't have anything transcripts related with this one, but you see that these three geomic builds, we actually have these different coordinates, and they all relate to the same canonical copy number variant, which just happens to have this ID. We can also search for the identifier itself. I'll copy this. It should get us back to the same thing. So instead of CAID, you can also search for the CACN ID. Yep, gets us back to the same thing. We also have this copy number variation overlap. So these for people who've worked with structural variants, they're often tricky in terms of how specific their ranges are and their locations. Sometimes they're estimates. Sometimes they have fuzzy ranges, unknown starts and stops. So this allows people to kind of start with a descriptor in this type of format. You can care about copy number or not. In this case, I'm not going to. I'm going to keep ignoring that for this. But for this specific range, we want to know what other copy number variants that are also in the same location. So we do this query here. You'll see that we have all these additional CACN IDs that exist within the same range. So we're going to keep extending this to figure out the most interesting and useful ways to display this information. So you can see that if a lot of these things differ by a single base, they're probably likely a very similar event. And we're going to keep getting feedback from the community to see like what makes the most sense for registering these and being able to query these based on locations. Like we talked about referencing to the physician, we can also do that for CACN IDs. So you can start off with a build genomic location. You can also do a search like this instead of using the descriptor method. And you see that we also get something very similar here. So this is basically also, you see everything in this URL here. Everything can be, this payload can be instructed like everything else for the short copy numbers. So you basically get your start and your end positions, your Manhattan distance, which is basically using these genomic coordinates to figure out how far away something is from the source or from the entity that you were curious about. Okay, I want to show what we have coming for non-CNVs as well. I have CNVs as well in here as well. But for these non-CNVs, what we're thinking structure-wise, and this is kind of just our first pass. So we're working on finalizing the structure and then extending the CNV part of the Allele Registry to support the non-CNV part. And just so you can see here some of these examples. So insertion often, from all the sources that we've looked at, does not include the actual sequence for these copy number variants. It's basically the size and the location, the size of the insert and the location that it occurs in. But when they have more information with the insertion, and we talked to the NOMAD folks about this, is they'll actually have the source of the insertion, which is often the same chromosome, but not always. So you can see we just have deletions, duplications, inversions, and then some of these complex entities. And not getting into the details here, but you can see that you basically insertion followed by an IDO, an inversion followed by a deletion. And so the main points here is you can see how we're basically looking at these fundamental units, the ones that are up top, and allowing us to build the complex entities by using this descriptor-based model. And this will hopefully get the ball rolling before we get more feedback from the community regarding some of these standardized models, in which we aim to support as well. So some of the next steps for the Allele Registry, we're aiming to support, always looking to add additional resources and also support different naming conventions, because different people should be able to get to the Allele Registry and then use that as a springboard to get different, either it be curation or just be interested about different metadata associated with these from different sources. So in that regard, we want to support the GA4GH variant representation specification, which is a digest, from from the observation that may become more popular in the future. I also want to automate some of the update processes, keep everything up to date, as quickly as possible. We're also aware of, different multi-reference down the road, with the pan-genome and support. So we have this on our radar of how is that going to affect the Allele Registry. Also, non-coding variant support. So we support non-coding, but not that, such things as, different splices, different other types of data sets. So non-CMV and complex structural variant support, which I just talked about. Also about the variant search tool, more of an integrated place where you can actually use as a jumping off point to find as much information as you can using the Allele Registry, Linked Data Hub, and other tools. Also, we want to add the preferred variant name and not just the ClinGen preferred variant name, as well as integration with the Linked Data Hub, which is Linked Data Hub is going to feed a lot of other tool sets down the road. As well as migration with link management. So one quick thing about the Linked Data Hub here, as you see in this top panel, this is kind of the current view we have of doing a search for the HGVS or the reference sequence position, gives you the canonical allele ID with a bunch of links and then genomic alleles, transcript alleles, things like that. We want to start extending the Linked Data Hub to do a couple of things. Maybe do some of this link management, which will be useful for other tool sets, things that use the Allele Registry, the Linked Data Hub, but also include these expansion sections that will have some of the interesting information about like in silico prediction scores, allele frequency, things like that we've excerpted from other data sets. So kind of having a place that has a lot of data in a single location, as well as links to the places that, that things that we didn't capture. So we're Linked Data Hub aiming to be the software infrastructure, kind of like the PubMed for gene and variant knowledge, kind of like a search for these types of things. So if we use the primary subject in the database as it's this canonical allele identifier, basically solving these problems that we've talked about, the Allele Registry allows us to kind of link everything kind of the centralized way, and also be able to curate based on seemingly the same event. And then producing low-level variant data to the VCI and other entities. So we want to have between the Allele Registry and the Linked Data Hub, we want to have this permanent reference for inclusion, for supporting evidence and variant interpretation, tracking of provenance, standardization of data, developing support for versioning of evidence. Yeah, so in summary, kind of introduced the Allele Registry, kind of defined the problem, so the Allele Registry was capable of doing. Talked about the current usage, existing functionality for small variants, also structural variant support plans for CMV and non-CMV, and then talked about the next steps. And then there's a lot of people to thank for all this work. A lot of people involved that are in our lab currently. Some people that were formerly in our lab. Too many people to list, but I just want to thank everyone for all the contributions and feedback, and it's all been very meaningful. And thanks again for the opportunity to speak before this group. I really appreciate it. Happy to take any questions.
That was great. Thank you so much, Kevin. Just an amazing amount of work has gone on into the Allele Registry, and it's such a fantastic resource. So thank you for going through all of that information with us and the demos. I do have a question from Megan, and Megan, please jump in if you want to add additional detail, but it sounds like they had been looking at a variant in TP53, I believe, and it looks like it's a variant where the last couple of nucleotides of an exon and into the intron have been deleted, and then there's an insertion. And she said they didn't get any information back on a frame shift for this variant. So I guess, does the Allele Registry, was this complicated by the fact that there's a del-ins that's kind of spanning the exon-intron boundary? I can give you the HGVS if that helps.
Yeah, that would be great to see that. So I believe we support the vast majority of HGVS expressions. And yes, so sometimes this gets tricky coming up with that, right? In terms of what you're actually deleting. And you're right, because that nomenclature of, do you have this minus symbol, or are you actually going into the intron? Are you deleting, or are you inserting into the thing past the exon, which exists in the transcript? So each case is a little tricky depending on what's available, what information is available. So if I say that HVS, I think we'll be able to figure out what we think is happening in terms of the observation and how we can actually then get that into the little registry, if even having to tweak the HUVS or something. Yeah.
Alrighty. She said also, yeah, tricky. Was there any functionality in the allele registry that we just didn't know how to use that could have helped us? Because it looks like she had figured out the correct HGVS, but yeah, and the interactively create or generate variant, that only looks at exonic sequence, correct? It doesn't look at intronic as well. It's just cDNA.
That's correct, and yeah, no, that's a very good observation. It's because it's just bringing in the transcript. That's a very good point, though, of how someone would be able to interactively generate something that, you know, I'm trying to think what that would actually look like. Yeah, so it should just be. Let's have a look at the transcript. I'm sure, like, oh, that's it. Yeah. So yeah, this wouldn't have.
Right. Yeah, and I guess this one's tricky too because, you know, it's deleting part of the donor splice site also, so maybe disrupting splicing. But yeah, it's a tricky one. So I put it in the chat. I don't know if it's possible to look at that. I don't know if you can see it,
Absolutely.
Kevin.
You have to, yeah, it's not letting me copy and paste from the chat. I'll take a screenshot and I think that'll, yeah. Yeah, I'll say that. Excellent. And maybe I didn't put it in, you know. And so I think we have emails and stuff like that, but for anyone wanting to contact me, I'll just put my email here. Sorry about that. I should have done that.
That's great. Thanks.
Yeah, and so yeah, again, feedback's always welcome, and you know, problems, and we try to get through these. So sometimes there's a lot of commonalities. If we see the same people, same type of questions being asked a couple of times, we work to address it. Some things are just one-offs, or sometimes there was a mistake maybe made in a manuscript. I mean, we found that a couple of times too, where something was actually, you know, copied and pasted incorrectly. We believe the variant was actually, you know, a single nucleotide before where it was listed, you know, things like that.
Okay. There are a couple of other questions too. Let's see. So one is that older transcript versions are not supported. I guess I've found this too. Like sometimes if you put in like 0.3 and the transcript's been updated to 0.4, then the allele registry won't, you know, it'll say, I forget what the message is, but you get some sort of error message. So sometimes if you just try the next decimal point up, it will work. So is there any, do you have any thoughts on that or any advice?
Absolutely, yeah. So what we do is we try to get everything that ClinGen has, and we never delete anything. So if ClinGen, you know, if ClinVars had it, for example, I'm just looking at to see when the last time this has been updated. Yeah, so it's 2019. So let's see. Yeah, so it really depends on what they have. So that was 2014. I was just looking at the one previous number before that. And so for whatever reason, the bulk transcripts that we download from ClinVars FTP doesn't have this 0.2. I don't know if it was marked a certain way, or they said it was unreliable, or if they do a certain date. So I've seen really old transcripts that we still have in the registry, but and other times, this 2014 one, I'm not sure why it's not available in the ClinGen data set. So we've explored a couple times. A couple times it wasn't present at FTP, but we found the following month it was present. So every once in a while we see maybe a glitch here and there, but we update monthly. So we have these protections in place to make sure that when we update, we're only adding, we're not deleting. So if they have it, we try to put it in there.
Okay, that's good to know. Thanks. And then here's a thanks for your wonderful talk, Kevin, from Allison. And she also is asking whether all short variants in large databases such as Nomad and ClinVar are registered with a CAID within the Allele Registry. That's correct, isn't it? So you are constantly updating from Nomad and ClinVar?
Yes. Yeah, I had to check the specific dates, but yeah, so everything that's in these sets should be registered in the Allele Registry. There are some cases where things are rejected, and that's because sometimes there's mapping problems between, let's say, GRCH 37 and 38. So there's a couple things that we see periodically that aren't in there, but we should be up to date up to Nomad 3. And whatever the most recent ClinVar releases. If anyone sees any problems, we can always explore.
Okay, I think those are all the questions. Are there other questions that folks have? We just have a minute or so left, but please feel free to unmute yourself or put a question in the chat.
I'm just curious. It's possible to imagine the situation when the small change would considered to be quote unquote copy number have been also entered the usually way, as for example, small deletion. Is it possible to imagine situation you would have two IDs, one CAID and another CACN ID for exactly the same change?
That is an excellent question, and we've had some long discussions about that. And you were absolutely right, because we've seen a lot of these structural variants that we would say, oh, we could easily create a CAID for that. So what do we do? Yeah, so our plan is never, once something's registered, it stays registered. We never go back and say, oh, that actually should have been the CACN ID. What I think what we're likely to do is probably to have duplication for entities where, let's say for Nomad, for example, let's say Nomad has the same resultant one in both of their data sets. In that case, you would have a CAID 1 and then CACN ID, X, whatever that happens to be. What we're aiming to do to solve that problem and down the road, and this is more just, we're still kind of whiteboarding this, is to have some knowledge that they're related entities. And we're either going to use the linked data hub or another type of entity to actually describe that and have something that says that, yes, we believe that these are either linked or they're the same event, or they seemingly look very similar type of thing.
Regarding legacy names. If you have, I know there are a number of genes where for years and years, there was a certain nomenclature used, and then they identified, I don't know, another exon or something like that. So the numbering has changed, but in the older literature, you're often seeing these, common variants that are labeled in one way, but now we know they have a different name and the old nomenclature was incorrect. Does the Allele Registry deal with that in any way with those legacy names, or is it really just only includes the current correct nomenclature? I think it's a question.
That's a great question. So if something was, incorrectly, let's say the position was wrong or something like that, and that it's registered and stays registered. That's something you would have to use a new thing. But for something like gene name or a transcript name, I'm trying to think of what nomenclature. Pull in the chat now. Let's see. Gene has outdated numbering. Is that a gene name, or is it referring to location?
Yeah, location, and often amino acid. I think this is what you're asking, Joanna.
I can specify. For example, for LDLR gene, the numbering of the amino acids changed from a while back. So the same variant that was amino acids change 41 is now called for 62, for example. So the numbering of the whole gene changed. And in older papers,
gotcha.
we have the PT41M, but we now know that this refers to what we now call it the PT62M, if that makes sense.
I believe, if I understand you correctly, so would the resultant, did the underlying sequence get updated? So what I'm asking, is it the same transcript for both of these?
What happened is the first 21 amino acids that are cleaved, they would not be counted before, and now they are. Yeah.
So that should result, that should be a different identifier, I'm guessing, for that sequence.
Okay.
In terms of what they provide for the protein. If I understand this correctly, in that case, you would basically, yeah, you would just have different, depending on where it actually, mapped to. So the way we kind of handle this is we kind of do on the fly mapping, in terms of generating the, the amino acid change. So if the coordinates are the same, if you started off with genomic or something with the transcript, it should result in the same, even if, because it would be based on sequence identity. I would have to look at an exact example to verify that, but that doesn't immediately concern me in terms of the underlying allele registry.
Okay, thank you. It's just that usually older papers only refer to the variants by protein numbering, and this is all we had. And that's our issue. But
So
thank you for your answer.
that would be, that's more difficult then, yeah. So then I think you'd almost have to have more information, because the allele registry would say, we're fine with whatever you want to say, but it has to be a valid change. So if you want to say that, you're valid, valid glue or whatever, it would say, well, if you don't say val in that location, it would say it's invalid. It wouldn't let you do that. So there's a little bit of, I guess, some checking on the allele registry end that said that if you say that the reference allele is a C, but it's actually a T, it won't let you do that.
Okay, thank you.
I don't know if people are still on. I'll just show it real fast. Let's see. Whatever, a number. I'll say A to C. Probably not going to be right off the thing. So yeah, good. So this is not a valid allele, and it very similar. So that sometimes that helps when something weird happens. Something changes. It'll say that's not correct. Well, that's outside of, I used the wrong sequence, but sometimes that'll help alert you to a problem. Yeah. So basically, I said it found the wrong one. So that's a little bit of sanity check. It's not perfect, right? You have 1 in 4 chance of it being, more or less, finding coincidentally finding single nucleotide. But
Okay, thank you.
Yeah, let me know if feel free to email me if I can follow up with that, perhaps some more details.
Okay, thank you.
Great, thank you.
Great. Well, thank you so much, Kevin. That was really fantastic to go through all of that information. And thanks to everybody for the great questions and discussion. And we'll look forward to talking with everyone again on February 11th. Right. Thanks so much and have a great day.
Great. Thank you. Thanks again, Jenny.
Thank you.
Bye.
Take care. Bye-bye.